Biostatistics is the application of statistics to a wide range of topics in biology. The science of biostatistics encompasses the design of biological experiments, especially in medicine, pharmacy, agriculture and fishery. The collection, summarization, and analysis of data from those experiments, and the interpretation of, and inference from, the results. A major branch of this is medical biostatistics, which is exclusively concerned with medicine and health. Almost all educational programs in biostatistics are at postgraduate level. They are most often found in schools of public health, affiliated with schools of medicine, forestry, or agriculture, or as a focus of application in departments of statistics. In the United States, where several universities have dedicated biostatistics departments, many other top-tier universities integrate biostatistics faculty into statistics or other departments, such as epidemiology. Thus, departments carrying the name biostatistics may exist under quite different structures. For instance, relatively new biostatistics departments have been founded with a focus on bioinformatics and computational biology, whereas older departments, typically affiliated with schools of public health, will have more traditional lines of research involving epidemiological studies and clinical trials as well as bioinformatics. In larger universities where both a statistics and a biostatistics department exist, the degree of integration between the two departments may range from the bare minimum to very close collaboration. In general, the difference between a statistics program and a biostatistics program is twofold. Statistics departments will often host theoretical/methodological research which are less common in biostatistics programs, and 2. Statistics departments have lines of research that may include biomedical applications, but also other areas such as industry, quality control, business and economics and biological areas other than medicine. The advent of modern computer technology and relatively cheap computing resources have enabled computer-intensive biostatistical methods like bootstrapping and resampling methods. Furthermore, new biomedical technologies like microarrays, next-generation sequencers for genomics, and mass spectrometry for proteomics generate enormous amounts of redundant data that can only be analyzed with biostatistical methods. For example, a microarray can measure all the genes of the human genome simultaneously, but only a fraction of them will be differentially expressed in disease versus non-diseased states. One might encounter the problem of multicollinearity, due to high intercorrelation between the predictors, in this case say genes. The information of one predictor might be contained in another one. It could be that only 5% of the predictors are responsible for 90% of the variability of the response. In such a case, one would apply the biostatistical technique of dimension reduction, for example via principal component analysis. Classical statistical techniques like linear or logistic regression and linear discriminant analysis do not work well for high-dimensional data, that is when the number of observations n is smaller than the number of features or predictors p, n less than p. As a matter of fact, one can get quite high R2 values despite very low predictive power of the statistical model. These classical statistical techniques, especially least squares linear regression, were developed for low-dimensional data, that is where the number of observations n is much larger than the number of predictors p, n greater than p. In cases of high dimensionality, one should always consider an independent validation test set and the corresponding residual sum of squares RSS and R2 of the validation test set, not those of the training set. In recent times, random forests have gained popularity. This technique, invented by the statistician Leo Breiman, generates a lot of decision trees randomly and uses them for classification. In classification, the response is on a nominal or ordinal scale, as opposed to regression, where the response is on a ratio scale. Decision trees have of course the advantage that you can draw them and interpret them, even with a very basic understanding of mathematics and statistics. Random forests have thus been used for clinical decision support systems. Gene set enrichment analysis (CIA) is a new method for analyzing biological high-throughput experiments. With this method, one does not consider the perturbation of single genes but of whole, functionally related gene sets. These gene sets might be known biochemical pathways or otherwise functionally related genes. The advantage of this approach is that it is more robust. It is more likely that a single gene is found to be falsely perturbed than it is that a whole pathway is falsely perturbed. Furthermore, one can integrate the accumulated knowledge about biochemical pathways like the JAK-STAT signaling pathway using this approach.